Every living cell in your body contains thousands of proteins, each folded into a precise three-dimensional shape that determines its function. For decades, scientists struggled to visualize these molecular machines in action. Then one researcher developed a revolutionary technique that would change biology forever. Wuthrich, a Swiss chemist and biophysicist, transformed our ability to see the invisible architecture of life. Born in 1938 in Aarberg, Switzerland, Wuthrich pursued his passion for understanding the molecular world, earning his doctorate in chemistry from the University of Basel in 1964. At the time, scientists faced a fundamental challenge. Proteins are too small to see with conventional microscopes, yet understanding their shapes is essential to comprehending how they work. The dominant method for determining protein structures was X-ray crystallography, which required proteins to be crystallized into solid form. But many proteins simply refuse to crystallize, leaving vast regions of biology unexplored. Wuthrich saw potential in a different approach. Nuclear magnetic resonance spectroscopy, or NMR, had been used primarily by chemists to study small molecules. This technique exploits the magnetic properties of atomic nuclei, revealing how atoms are connected and positioned in space. Most scientists believed NMR could never work for something as large and complex as a protein. Undeterred, Wuthrich spent years developing new methods to apply NMR to biological molecules. Throughout the 1970s and 80s, he pioneered techniques that could extract structural information from proteins in their natural state, dissolved in solution rather than locked in crystals. This was revolutionary because proteins in solution behave much more like they do inside living cells. The breakthrough came through painstaking innovation. Wuthrich developed systematic approaches to assign NMR signals to specific atoms within a protein, then used the relationships between these signals to calculate distances between atoms. By combining thousands of these distance measurements, he could reconstruct the three-dimensional structure of an entire protein molecule. In 1984, Wuthrich and his team published the first complete protein structure determined by NMR. The achievement opened a new window into the molecular world. Suddenly, scientists could study proteins that had resisted all previous attempts at structural analysis, including membrane proteins and flexible proteins that play crucial roles in cellular signaling. The impact extended far beyond academic curiosity. Understanding protein structures in solution revealed how these molecules move and change shape, providing insights into their biological functions. Drug designers could now see their molecular targets in unprecedented detail, leading to more effective medications. Researchers studying diseases caused by misfolded proteins gained new tools to understand what goes wrong at the atomic level. Wuthrich's contributions earned him the Nobel Prize in Chemistry in 2002, which he shared with John Fenn and Koichi Tanaka for their work on biological macromolecules. The Nobel Committee recognized that his methods had become indispensable tools in modern biology and medicine. Beyond his technical achievements, Wuthrich trained generations of scientists who carried his methods around the world. His laboratory became a hub of innovation, constantly refining and extending NMR techniques to tackle increasingly complex biological problems. Today, the structures of thousands of proteins have been determined using the approaches he developed. The elegance of Wuthrich's work lies in its fundamental principle: observing molecules in conditions that mirror their natural environment. While X-ray crystallography provides exquisite detail of frozen structures, NMR spectroscopy captures proteins in motion, breathing, and flexing as they do within living cells. This dynamic perspective has proven essential for understanding how proteins actually function. Wuthrich transformed protein structure determination from an art practiced by a few specialists into a systematic science accessible to researchers worldwide. His vision and persistence gave humanity the ability to see the molecular machinery of life with clarity never before possible, bridging the gap between chemistry and biology in ways that continue to yield discoveries decades later.